Cool. So thanks everyone who's tuning in for tuning in. You might be expecting to hear about the functional hierarchy of choice, but due to a scheduling change, you're going to be stuck with me. I definitely encourage you though to check out the work by David from our lab. It's posted up on bioRxiv and it's super exciting. But today I'm going to be talking about our work decomposing natural behaviors in freely moving macaques. Our ultimate goal though, like most other people's in our field, is to understand how the brain generates behavior. And the way we typically study that is mapping brain activity to behavioral output. We put a monkey in a chair and let him perform some task. And this has led to a lot of powerful insights for sure, but the behavioral repertoire that can be expressed is going to be severely limited. And this is a shame because we are throwing out a lot of key information, namely what their body movements can tell us. And consider how much information we can infer from the body language in this picture. We can tell that the guy is trying to engage the lady in conversation. He's clearly interested in her, but she is not. But if she changes her pose and now she's looking at him, well, now all of a sudden we can infer that, okay, she's interested, or at the very least, she's willing to engage with him. There's an important principle here. The expression of behavior can change independent of what the physical space is. And it's going to depend on what is relevant in that moment, what is being rewarded, what is the time scale over which we're being rewarded. And more than that, we can infer another person's internal state or motivation or intentions from the expression of their poses. And this gives us an important way to then study how the brain can organize and instantiate intentional behaviors. Now, we study this problem in macaques, as they offer a really rich source of neural data and while still having a really high translational capacity for humans. And our ultimate goal is to understand how the brain generates self-generated behavior. But to do that, we need a large corpus of behavior. Now, for various reasons, one of the best ways to do this is by immersing our subjects in large environments where they are free to move around. And this gives them the freedom to enter many different postural states over the course of an experiment. This is the setup that we're using to tackle these sorts of questions. It's a large enclosure where the monkey is free to move around naturally in all 3 dimensions, and we record them with 62 synchronized cameras. And that's important because then that allows us to accurately reconstruct their pose as they traverse through their space. And you can see the results of that reconstruction in this video. So on the left is a video of the monkey from 4 different camera views, and the colored lines are the reconstructed skeleton of the monkey, where we're tracking key joints like the neck, hands, head, or tail. And as you can see, we can track the monkey very well, even when some of the camera views have occluded joints or don't even capture the monkey. So with this setup, what we're going to do is then ask our monkeys to perform different tasks. So one of the tasks is a really classical foraging experiment task with depleting rewards. So the monkey has 4 different feeder boxes. Each feeder box has five rewards. Once one feeder has been depleted, there's a really large timeout. So the monkey, if it wants any more reward, it has to go to some of the other feeders. We also had one where we had a variable schedule. So some feeders had the same sort of structure with depleting rewards. Other ones offered a really large payout, but at a random time. And finally, as a control, we had a condition where there was no task at all. So same physical space, the feeders are all up, but they're not doing anything though. So the monkey's free to move around and do whatever it wants to do. So in total, we collect 18 data sets. So that's 2 subjects, 3 tasks, 3 examples from each. And we're going to split each data set into 10-minute segments to sort of trialify the data. So to get a holistic picture of the kind of stereotype behaviors that the monkeys perform in this cage, we used a dimensionality reduction technique called UMAP, Universal Manifold Approximation and Projection. So we take our high dimensional matrix of joints, reduce it via PCA to improve the computational time for UMAP, and then let UMAP learn a lower 2-dimensional embedding. And then you can think of each UMAP axis as something like a PCA axis. It tries to capture the global structure, the structure of the data rather. But the really good thing about UMAP is that it can find these embedding dimensions that can balance both local and global structures. And this is unlike, for example, t-SNE, which in a way exaggerates the differences between any local clusters to make them appear really far apart. So from this UMAP embedding, we can then generate a probability density map of each pose. Peaks in this map are stereotype behaviors that are going to occur often. And we leverage this and to use a watershed segmentation algorithm on this density map to cluster the data into iconic poses. So here, for example, we found 2 iconic poses. With all that said, this is what our embedding space actually looks like. Each of the peaks here represents an iconic postural state that is going to be visited often. And I've shown here a few examples of this. So for example, here on the left, we see the monkey's leftwards and rightward stride as it's walking. And it spends a lot of time walking, right? But there's other states where we look at it and we know exactly what they mean. They're semantically meaningful. For example, when the monkey is sitting, or when the monkey's standing and it's kind of looking out outside the cage, out into the world. When the monkey's climbing along the wall, or even when it's hanging upside down. So overall, on average, our states last about 3/4 of a second. And so really this is a sub-second decomposition of behavior that we've performed. But we can get even more information from this embedding space. And we can see this by looking at the transition probability matrix. So this is the probability of going from some particular state and ending up on some other state on the next time point. And I've reordered these according to the distance of these states on this map, right? And generally speaking, what we can see is that most transition happen to nearby states. We see this sort of blocky structure in the data. So that means, for example, if the monkey's spending time walking, it's going to move to another walking state often. But there's even more that we can take out from the transition probability matrix. So one expectation that we naturally have is that monkey's behavior is going to be goal-directed, or at least intentional. We can reframe that question and ask, are there hidden states that allow us to predict monkey's behavior far into the future? And we can do this by asking if the transition matrix is actually Markovian. Now, this analysis is lifted pretty much from Gordon Berman's 2016 paper. They've done really phenomenal work decomposing the behavior of fruit flies. Now, a Markovian system is going to be fully described by the transition probabilities from the current state and then the subsequent state. So if the system is Markovian, if we multiply this matrix by itself X number of times, representing X number of transitions, we're going to get something like this, which represents the steady state probabilities of each state. And you can see there's pretty much no information left, or no structure left, sorry, in this matrix. Now, another feature of a Markovian system is that our ability to predict far into the future decays exponentially. And this is evident when we can extract the eigenvalues of the transition matrix after X number of transitions, and we get a plot that looks something like this. So here I've plotted the absolute value of the maximal eigenvalue as a function of the number of transitions, and you see an exponential decay, in the eigenvalues. But the real transition probabilities actually look something like this. There's still a lot of structure in here. And when we plot the decay rate of the eigenvalue, we can see it actually decays much more slowly. And so what this implies is that there are hidden states that have a memory and that would allow us to predict the monkey's behavior far into the future. But to put it all together, what this also means is that the monkey's behavior is Markovian at short time scales and non-Markovian at longer ones. But in some ways, this is not surprising. It makes sense. If I try to predict, for example, where your arm's going to go in the next time step, I can do it really well for the next time step. But if I want to predict far into the future, I now need to have information about your goals, or your reach targets, and things like that, the hidden states that we can't observe. But we see the same sort of decay behavior for many of our top eigenvalues, 6 of which I've plotted here. And you can, when you compare every colored line, the dashed versus the solid versus the dashed line, we can see that the real data has a much slower decay. And so what this actually tells us is that there are multiple time scales of behavior for our macaques. But this excess decay, the difference between the Markov expectation and the observed rate of decay, does not differ by task. And so this is obviously kind of surprising, and it tells us that the task does not influence behavior in terms of the timescale of predictability. But this does not mean that behavior is actually devoid of context information. So even though every state is likely to be expressed, the relative frequency of behavioral expression is going to be subtly different. And so we can get at that by looking at the behavioral expression profile. So this is the frequency with which a monkey is going to visit any particular state. And this kind of approach is has been pioneered by Bob Datta's lab, who've also done a lot of really great work in decomposition of mouse behavior. And recently they actually published a paper in Nature Neuroscience looking at this expression profile as a function of drug conditions. So what we're going to try to do is decode the task as a function of this behavioral expression profile. And indeed, we can actually predict which task generated this data with 76% accuracy, above what we'd expect from a random shuffle of our data set. And we can also predict the subject with 99% accuracy. So our subjects are extremely idiosyncratic in how they move and express their behavior. And with that, I'm ready to conclude. So we found that we've leveraged a method, postural embedding, that can actually preserve meaningful semantic structure in our data. And we found that the behavior is Markovian at short time scales and non-Markovian at longer time scales. So sort of giving us now a handle on, starting to give us a handle on the hidden states that we can leverage to ask questions about our neural data. The predictability of behavior does not depend on the task, but the actual expression of behavior does. And so these kinds of results are really a really crucial stepping stone to the kinds of questions that we want to ask moving forward, where we want to look at more fine-grained analysis of poses that can predict the task, relate behavior to the actual different task elements themselves, and finally actually map all this to the neurons. And so with that, I'd like to thank everyone who's been really critical in this project. It's a really large project involving a lot of people in the lab. My principal advisors, Jan Zimmerman and Ben Hayden, been a fantastic source of support. Hyunsu Park, who's a collaborator with us, has been really critical in actually getting the pose deconstruction done, as well as Praneeth Bala, David Basin, Jen Holmberg, and Runal Zambre, who were also involved in the day-to-day operations of the cage. And with that, I'd like to thank you all for listening.
Great, thank you very much, Ben. We went a little bit above time, but it's all right. On behalf of all the attendees, I'm gonna have you give you a sort of round of applause. Yeah, very nice talk. And it seems, yeah, I mean, I think there's a collective attention more and more in the field towards understanding better and better and quantifying in more details behaviors. As you mentioned yourself in rodents and now monkeys and then I guess also humans, if we want to understand better the neural circuits. Yeah. So I don't want to take so much time, so I'm going to ask the first question that we have from the audience. Malcolm is asking, is the eigenvalue decay rate at all dependent on how many UMAP dimensions you use? Have you tried more than 2, for example?
Yeah, that's actually a great question. I've got a figure for this. So one thing that we can do is a sort of naive reclustering. Instead of, we take our 31 states, our embedding space, and then try to recluster it into different numbers. This is kind of what the clusters look like. And then we look at the excess decay, right? And what we see is actually it pretty much doesn't matter. Only if we get down to 3 clusters or lower will we get a difference in decay rate, or a smaller difference in decay rate. But still, it's above chance. So it's still non-Markovian, but it does look to be a little bit faster with less clusters.
All right, great, thank you. I had one more question. I mean, again, maybe a basic one. I think you mentioned that briefly, but I did not really capture that. So what is the difference between these UMaps and the t-SNE maps, and what are the advantages of one representation over the other?
Yeah, well, so what t-SNE does is it exaggerates differences. So if there's going to be natural clusters in the data, what it's going to do is actually try to pull them apart as much as possible, so that you can visualize and analyze them better. But that means that we don't have a way to actually map distances, like distal clusters, right? The distance between them is not a meaningful number. And so UMap actually has a parameter that lets you balance the sort of preserving the local structure versus the global structure. So it gives you a more kind of, I guess, holistic view of what the actual data space looks like.
All right, great answer. Thank you very much.